Hello everyone. A very good evening to all of you. Can you hear me everyone? Is the screen is visible to all of you? Good evening Amit. Good evening Sunshine, Anusha. So how is the Power Hour series going on? I know today is the last day, but still, how was the 4 days of the CSIR NET Power Hour series? I guess it might have helped you a lot in revising the concepts, understanding the way we need to approach a question, right? Yes. Good evening, Minakshi. Good evening, Arzu, Prashant, Anusha, Red. Okay. Okay. So, today being the last day of this series, so we are taking the two most important, like one of the two most important topics or units, I can say, Unit 2 and Unit 4. Well, before getting into these two units, one thing I wanted to say you that this is completely a kind of units from where you can get direct questions. And at the same time, you have to have some practical knowledge with respect to Unit 13. So when we enter into Unit 4, there you'll find some conceptual, analytical, even practical based questions. So overall speaking, these two units are very important from examination point of view. I guess you all are aware of this fact. So without wasting much time, let's get into the session. So we will begin with Unit 2 first. Okay, so here is the first question. A membrane associated protein is composed of 7 alpha helices, with each helix containing 19 hydrophobic residues. While treating the membrane with all kinds of proteases, a major portion of this protein remains intact. Even after treatment with high salt, buffer, pH 5, the protein failed to dissociate. Now you have to predict the appropriate nature, orientation of this protein in the membrane. So the first option is peripheral glycoprotein, integral protein with 7 membrane spanning regions, peripheral protein with both N and C terminals exposed to outer surface of the membrane, or cytosolic surface of the cell membrane. So you can give your answers. See, basically, membrane based questions you can always expect either in the form of part B or in the form of part C. Whenever it comes in the form of part C based questions, usually what happens is that they are a bit challenging. Of course, if you reread it and if you have that concept in your mind, no one can stop you. Yes, I can see many answers coming up. Is there anyone else would like to give your answers? Very good. Suchitra, Kavya, Red, Sunshine, Gayatri, Soumya. So never miss this topic from your syllabus. As I told you, you can expect either in the form of part B or in the form of part C. Okay, fine. So the correct answer, as you all mentioned, absolutely correct. It is the integral protein with 7 membrane spanning regions. Well, in the question itself, they have clearly mentioned about 7 alpha helices. So with this topic, with this particular point itself, you can note down 7 membrane spanning regions. Second concept over here is you should be aware of difference between a peripheral protein and an integral protein. You know that integral protein is the one which is tightly attached to the membrane. So you need to provide with specific detergents that will interfere with the hydrophobic interactions, then only the integral proteins will be extracted. So that's the reason option 2 is the correct answer. As you can see in this image, this is our fluid mosaic model, the model for plasma membrane. We are having Peripheral membrane protein, as I told you, it is present in the periphery of the membrane. If we speak of integral membrane protein, they are spanning within the membrane, either single or multiple. Apart from that, you will also find some lipid anchored proteins being present. Okay. So this integral proteins can either exist as single transmembrane helix or it can exist as multiple transmembrane helix. And it can also exist in the form of beta barrels, that is transporters, channels that will allow the movement of ion molecules. Peripheral, so you can identify how it works, right? Now, focusing on integral membrane proteins, we have all total of 6 different types of integral membrane proteins. Now, how these integral proteins are classified, any idea? We have 6 different types of integral membrane proteins. Any idea how it is being classified? Like based on something, right? It is based upon, if you look into this image, it is based upon the orientation of the protein. If you see, the type 1 is basically the one whose N terminus will be exposed to outside. Can anyone give me an example of a protein that will have this kind of protein whose N terminus will be outside? Exactly, Manoj. The position of the amino group and the carboxyl group. Absolutely correct. So can you give me an example of this type 1 where you will find N terminus is present outside and C terminus is present inside? You'll find glycophorin. Right? Now, exactly opposite is your type 2, where you will find N-terminus orientation is towards inside. And here you can take the example of transferrin receptor. Okay. Next, moving on to the type 3. This, you can say, a single polypeptide with multiple transmembrane helices. Can you give me an example of this? There are a number of examples which comes under the category of type 3. Can anyone give? Very good. It is your GPCR. That means, yeah, GPCR means single polypeptide but is having different transmembrane helices. Exactly. You are having CFTR as well. Then you are having GLUT transporters. So that all comes under the category of type 3. Now, what is the difference of type 3 to type 5 is that here you will be having individual proteins accumulated together forming a channel. So, what should be an example over here? Individual proteins accumulated forming a channel. Here, single polypeptide that is having the transmembrane helices. But here, multiple proteins accumulated forming a channel. Which one it could be? Very easy. Gap junctions. Are you aware of gap junctions? Have you heard of gap junctions? Not tight gap junctions, we can say. Why gap junctions? Any idea? Because we have 6 connection proteins that accumulate in the form of a channel. Right? So that becomes your type 4. Type 5 is lipid anchored. So you can easily take the example of GPI anchor. Then type 6 is a protein that should have a transmembrane helix plus a lipid anchor. So far, I did not find any example for this where the transmembrane helix and the lipid anchor both will be coming into the same time. Okay. So moving on to the next question. Next question is lipid raft. Lipid raft is also one of the famous, common topic when we speak of CSIR exam. So lipid rafts are rich in both sphingolipids and cholesterol. Cholesterol plays a central role in raft formation since lipid rafts apparently do not form in its absence. So why do you think cholesterol is essential for the formation of lipid rafts? Because cholesterol decreases the mobility of sphingolipids in the lipid bilayer. Large head groups of sphingolipids tend to repel each other, or it interacts with fatty acid tails in the membrane, or the planar cholesterol molecule can fill the voids that form underneath the large head groups of sphingolipids. What should be the reason why cholesterol is required for lipid raft formation? Again, this is also similar to your previous topic where you can expect both in the form of 2 marks as well as in the form of 4 marks. I think in the last exam, we had this question in the form of 2 marks. Can anyone give the answer for this question? What is the necessity of cholesterol in the lipid raft formation? Okay, I can see Pratik has given the answer. What about others? Any idea? I can see miscellaneous answers coming up. Okay, let me give you the answer then. Here, the correct answer becomes 4. Why it is 4 means the planar cholesterol molecules are postulated to fill the voids that form underneath the large head groups of sphingolipids. That means what? See, this is our lipid raft, right? You can see glycosphingolipids are present. These are your transmembrane proteins. So if you take the entire thing, we call it as a lipid raft. You can see between the sphingolipids and the membrane proteins, gaps are present. So these gaps are filled by your cholesterol molecule. Okay. That is the reason 4th answer is the correct answer. And you can see that by filling the voids and increasing the packing density, cholesterol contributes to stability as well as organization of lipid rafts. I hope so this question is clear to all of you. Or is there any confusion? Meanwhile, I just wanted to ask you one more question. What are the 2 most important interactions which you can find within the biological membrane? Like if you take into consideration the phospholipids, how are they interacting with biological membrane? Do we have any specific interactions? Even based upon this interactions also questions come in the exam. Very good evening, Manjoy. Yes, one interaction could be your hydrophobic interaction. What will be the other one? Absolutely correct. Manoj, Sunshine and Kavya. Apart from hydrophobic, which one will be the other interaction? It should be hydrogen bonding. Hydrophobic and hydrophilic are the nature of phospholipids. Like this is philic and this is phobic. But interactions with the membrane basically comes from your hydrophobic and hydrogen bonding. You might be aware that cholesterol is having OH group. So this OH group interacts with your head of the phospholipids. Similarly, tails will be having hydrophobic interaction. Clear to all of you, or is there any confusion? So be clear with the composition of lipid rafts, properties of lipid rafts, types and their differences. Okay. Next question. Now this is based upon the membrane fusion with respect to snare protein. See, basically when you go for unit 2 and unit 4, you'll always expect statement based questions or match the following kind of questions. Okay. So looking into the snare proteins. So these are the few statements provided to you. The snare transmembrane protein exists as complementary sets. V snares on vesicle membrane and T snares on target membrane. If you look into the structure, V snare is made up of 3 proteins and T snare is made up of single polypeptide. So this V snare and T snare will interact with each other, forming a 2 helical bundle. Membrane fusion is catalyzed by the energy that is freed when interacting helices wrap around each other to pull the membrane faces together, and whatever the water content will be there, that will be released out. Identify the correct combinations over here. Okay, I can see Lakshmi has come up with an answer. What about others? This is also a topic from where 4 marks questions are basically expected. A part of, I can say, solute transport, where membrane fusion plays a very important role. Okay, I can see further answers coming up. Very good. Okay, I can see many of you are giving the correct answers. Great. Okay, so let's look into the answer now. So the correct answer over here is 4. Now, why it is A and D? Because if you see, V snares are something that is made up of a single protein, whereas T snare is something that is made up of multiple protein subunits. If you see, this is our T snare that is having syntaxin and SNAP25. And from the vesicle, you are having synaptobravin. So this is how the interaction takes place. And this clears why option statement B is incorrect. Now, why statement C is incorrect? Because it does not form double helical bundle. Rather, it forms a 4 helical bundle. How? You know that we are having interaction of V snare to T snare proteins. So each of these proteins will be having a snare motif that is made up of an alpha helix, approximately 60 amino acids. So they will interact with each other, coil-coil interaction, and they will form an heptad repeats. That means a repeat of 7. Okay. Now, who is contributing this 4 helical bundle? You'll be having VAMP, or you can also say as synaptobravin. Then one helix can be contributed by syntaxin, and other 2 will be contributed by SNAP25. So all total you got 4, right? So that helps in our membrane fusion. Next question. Find out the incorrect match among the blood group. So normally we have a tendency that when we read the questions, we go on very quickly. We forget whether it is mentioned is incorrect or correct. So always read the questions thoroughly. Find out the incorrect combination from here. I hope so. Whoever has given the incorrect answer are able to understand why you went wrong. Which one is the incorrect answer? Yes, again, I can see many correct answers are coming up. Absolutely correct. Yes, very good. So the correct answer over here is 4. Now, why 4 is incorrect? You have to understand this. So this is our O antigen, which is composed of these many sugar molecules. That is galactose, N-acetyl glucosamine, galactose, glucose, and one galactose connected to fucose. So in this O antigen, if you are adding N-acetyl galactosamine transferase, you are getting A antigen. And if you are adding galactose transferase, you are getting the B antigen. So you are having one extra galactose here and one extra N-acetyl galactosamine here. So that is what the exact formation of blood group antigen. See, the carbohydrates of glycolipids determine whether a person's blood group is A, B, AB or O. So depending upon the addition of sugars, the blood group is determined. Okay. Can you identify the A, B, C? What does it indicate? Now this is the part of a intestinal epithelial cell. You need to identify what does A indicates, B and C indicates. Which transporter it is. I hope so you are able to recollect whatever you have studied so far. And you are able to apply it over here. Yes, it's a symporter. Absolutely correct. Very good. Exactly, Anupam. Yes. I can see some of you has given the incorrect answers. No issues. See, why did you went wrong? I'll explain you. So the correct answer over here is answer 1. That is A indicates GLUT5. Fructose transporter. Do you remember everyone? So that becomes your GLUT5. This one moving into the blood from the intestinal lumen moving into the blood, that is carried out by GLUT2. Now this is an example of a symporter, sodium glucose co-symporter. So that becomes your SGLT1. Okay. So for that you are having specific tables of glucose transporters where you are given provided with different GLUT transporters, like 1 where it is expressed, 2 specifically in liver, pancreas and intestine. So intestine is there. That's the reason you got the answer over here. Right. Next, you are having 4, that is muscle, fat and heart, 3 exclusively in brain, and 5 is considered to be a fructose transporter, specifically present in intestine, testis, kidney and sperm. Okay. Next, there are some examples of co-symporter like SGLT1. Other than that, sodium phosphate co-transporter is there. Iodide symporter is there, sodium potassium chloride and potassium chloride co-transporter. So all these transporter are expressed in different parts of our body. Similarly, we are also having antiporters like sodium calcium exchanger. I guess you are aware of this because this is one of the important exchanger when we speak of cardiac action potential. Then sodium hydrogen exchanger exclusively for kidney. This is your AE, anion exchanger. You might be aware of chloride shift and all from unit 7. So that becomes your this one. Okay. Next, a diabetic patient. So based on that, you have another question. A diabetic patient has a high blood glucose level due to reduced entry of glucose into various peripheral tissues in addition to other causes. There is no problem of glucose absorption, however, in small intestine of these patients. The following statements are put forward to explain this observation. Glucose is transported into the cells of muscles by GLUT transporters influenced by insulin receptor activation, or the glucose transporter is mediated by SGLTs not dependent on insulin, or by facilitated diffusion or secondary active transport occurs in muscles. Now here also you have to identify the incorrect statements. Find out the incorrect statements from here. Just if you understood the previous question, definitely you can correlate this over here. Yes, absolutely correct, Arvind. Sorry, you have given this answer, right? Okay. Okay. I can see miscellaneous answers coming up. You have to identify the incorrect statement over here. Read the question carefully before giving the answer. Incorrect statements. And I can see many of you have clicked on for like given the answer as correct statement. Some miscellaneous answers are coming up. Let's check out the answer, what exactly it is. Okay, still some of you has given the correct answers. Very good for that. Correct answer over here is 4. That is C. Undoubtedly, this is the correct answers. Glucose is transported into the cells of muscles by GLUT transporters. Right, whenever an insulin goes and binds to the receptor, so GLUT transporters will be relocated to the membrane, taking up the excess of glucose. Similarly, just now we have discussed in the previous question where sodium glucose co-transporter is working, and that is not dependent on insulin. So if you are confident that A and B are correct, obviously C and D are just opposite to that. So obviously you can eliminate these two options, or you can say mark it as a answer. Because when you are confident that it can go for GLUT transporters, how can you expect for C? And if you have already given as GLUT transporters and sodium dependent glucose transporters, there is no requirement of secondary active transfer, because secondary active transport is against the concentration gradient that takes the help of primary concentration gradient. Right? Next question. Are you aware of Clathrin vesicle formation, Clathrin coated vesicle formation? Are you all aware of how a Clathrin coated pit is constructed? If so, you have to identify the correct sequence of events that is responsible for making this Clathrin coated vesicle. And this question came last year. Last year means the last exam which came, 2022, which was held recently. Okay, that's good, Hemlata. Kindly proceed with the answer. Receptor ligand recognition and binding, recruitment of adapted protein, vesicle formation, uncoating of Clathrin coats. Very good, Sriyoshri. Red. Clathrin coated vesicle formation. Yes, absolutely correct. I can see all of you are giving the correct answers. Ajina, Hemlata, Swagat, Akhil, Arzu, Soumya. Very good. So you are aware how a clath, and this was a 4 marks question, came recently. Just imagine how easy it is. See, A, B, C, D. The same sequence is being used. Okay. Now, this is a Clathrin triskelion. So basically, why you call it as a triskelion? Because they have 3 heavy chains, 3 light chains with 3 globular heads. That is for attachment. Here you are having the adapter proteins. So adapter proteins are having different subunits. These two combine together forming a lattice. Okay. And how they are forming a lattice? The mu chain which is present over here will be connected to the receptor. The beta chain will be connected to the Clathrin. And how this adapter will be recruited to the membrane? With the help of a lipid molecule known as PIP2. Okay, this is how the receptor to clathrin coated pits is being connected. Now, based on this, I have another question for you. This is regarding familial hypercholesterolemia. Now, you have to identify what is the cause of this particular disease, familial hypercholesterolemia. So, you know that receptor mediated endocytosis, apolipo B on the surface of LDL binds to the corresponding receptor. Coated pits will be constructed, internalized by endocytosis, traffic to lysosomes, and finally recycle. Patient reports this particular disease. Now, what is the reason behind it? Mutation in the LDL molecule, defect in the LDL receptor recycling, mutation in LDL receptor, or defect in cholesterol binding to its receptor. What is the correct reason behind familial hypercholesterolemia? Absolutely correct, Anupam. Try on. Now, this is based upon the previous question. This is also a 4 marks question, came some years back. Absolutely correct. Ajina, Arzu, Soumya. Okay, very good. I can see everyone is giving the correct answers. Absolutely correct. So, always remember the LDL receptor is having a conserved amino acid sequence known as NPXY. If any mutation occurs in this conserved amino acid sequence also, your LDL will not bind to the corresponding receptor. So in that case also, the disease arises. Okay. So this is how it works. You have LDL bound to the receptor, clathrin pit sphere constructed, pinch off from the membrane, forming a coated vesicle, enters inside the early endosome, late endosome. You have got low pH, contents will be separated, and the LDL will be released into the lysosome, where it is recycled and utilized by the cell again. Okay. Next question is regarding autophagy. Autophagy occurs when cell contains aggregated proteins. Autophagosomes fuse with any organelle. It's a single membrane structure, autophagosomes, and it can fuse with lysosomes forming autophagolysosome. You have to identify the correct combination of statements. So basically from unit 2, calculation based questions are very rare. In fact, I can say there is not much being asked in the exam. Even if it comes also, maybe rarely it comes under microbial physiology. But that also recently, if you check the question paper, there we come across a question from match the following. So I have put that question also just to make you aware. Okay, we'll come to that. Yeah, what is the answer over here? Which of the combination is the correct combination? So here they are asking you the correct combination. Again, miscellaneous answers coming up. Students, are you getting the things cleared? Like, I mean, whoever is giving the incorrect answers, when you are able to understand why your answer is incorrect after my explanation. You are able to follow, right? Okay. Fine. Let me show you the answer. I can see some miscellaneous answers coming up. So the correct answer over here is A and D. Autophagy occurs when cell contains aggregated proteins. Autophagosomes fuse with lysosomes to form autophagolysosome. Now, why this statement is incorrect? Because it will not fuse with any organelle. It will only fuse with lysosome. It is not single membrane structure. It is made up of double membrane structure. Okay, that is the mistake over here. So if you see, this is our phagophore. So many phagophores accumulate together forming an autophagosome. Now this becomes a double membrane structure. Fuses with lysosome. So lysosome is having hydrolytic enzymes. So it will release the contents and degrades it. Okay, so that is the process. This is the same thing being mentioned. Next question is regarding Kedal receptor. Can anyone tell me what is the significance of Kedal receptor? Like it is the signal sequence for which cell organelle? Before going to this question, may I get an answer for that? Yes, it is the signal sequence for endoplasmic reticulum. Very good. Now, find out this question. Kedal receptor is found in COF1 vesicle. Affinity towards Kedal sequence is high at higher pH. Synthesized on the ER, is present both in ER and Golgi. So you have to identify the correct combination regarding the Kedal receptor. Lakshmi Gupta, so quick in giving the answer. So quick, everyone is giving the answer. Okay, there is some confusion over here. There is a confusion between option 3 and 4. As I can see, majority of you has opted for either 4 or 3. Okay, let's find out how does it works. The correct answer over here is A, C and D. For this, you need to understand this table. See, whenever you go for unit 2 protein targeting, tables are your saviors. Because if you remember the table properly, you can score 4 marks out of it. Because this is an example of a 4 marks. COP2 vesicle is coming out of your endoplasmic reticulum, moves to cis Golgi. Now this is considered to be forward movement. Or you can also call it as an anterograde movement. COP1 is moving back. That means cis Golgi to ER. That means it is a reverse order. You call it as a retrograde. Clathrin and adapter can move to different locations. Either it can go from trans Golgi to endosome or plasma membrane to endosome. Depending on that, you are having the coat proteins present and GTPase present. So if you see only for COP2, you are having SAR1 as the GTPase. That is the energy required for movement. But for remaining GTPases, for remaining vesicles, you are having ARF. Okay, easy way to remember. Next, what it is asked, Kedal receptor is found in COP1 vesicle. Why? Because Kedal is a signal of retrieval sequence. Retrieval sequence means by mistakenly if a protein enters into the Golgi, it has to be returned back to the ER. For that, the sequence is there. So basically, it is for soluble proteins. For membrane proteins, you are having KKXX. So that option is correct. Next, this we don't have any proof of this, but it is synthesized where? It is synthesized in the ER. Right? But it can also shuttle between ER as well as your Golgi. So that's the reason A, C and D are the correct answers. Okay. Next. Cytoskeletal proteins. Now, these are accessory proteins of microfilaments. You have to identify the correct combination. I hope so. The confusion between 4 and 3 is clear. to all of you? Yes, very quickly gave the answer. Jiva. And on this aspect, I just wanted to remind you one more thing. You have got 3 cytoskeletal proteins: microfilaments, microtubules, intermediate filaments. Remember their properties, drug inhibitors, as well as the functional difference. Okay, try to remember that. That is important from examination point of view. And in the recent past, if you look into the question paper, majority of the questions are based upon this accessory proteins. So you have to be very clear with accessory proteins. Any idea regarding this, any one of you? Because I have seen no one is answering excepting Jiva. Or shall I say you the answer? If you find it complicated, we'll explain you regarding both microtubules as well as microfilaments. Coffeeline, propylene, thymosin, formin. How are they connected to column 2? Okay, I can see Anupam gave the answer. Confusion. Don't worry. I will remove your confusion sooner. Don't worry, we'll explain you. Lakshmi Gupta. Sure, dream narrator. Okay, I can see many of you are not able to get it. Fine, no problem. So the answer over here is 3. Okay. We instead of looking over here, I will directly show you the image. First, we will look into the accessory proteins that are present for microtubules. Okay. Whoever has given the answer as 3, very good. Congratulations for attaining a 4 marks out of this. Whoever is finding it difficult, just have a look over here. So microtubule associated protein, you are having gamma TERC, gamma tubulin ring complex. You have to be very careful. It binds in which direction, whether it is polymerizing or depolymerizing. So this is responsible for nucleating the assembly and remains associated with minus end. Okay, towards the minus end, they are carrying out their activities. Stat mean binds to the subunits, preventing the assembly. That means depolymerization. Plus tips, they are associated with the plus end and help in connecting to the membranes. That means they help in polymerization. Yes, Anupam, you are correct. Kinesin 13 plus disassembly. XMAP 215 plus end but assembly. So see, these two are contradictory to each other. This is disassembly, this is assembly. Both are acting on which portion? Plus portion. Catenine breaks down. MAPS, that means microtubule associated protein, stabilizes the entire microtubule. Then these are some other cross-linking and filament bundling proteins which comes under microtubules like tau, MAP2, plectin, etc. So this is your microtubule associated proteins. Microfilament associated protein, you are having formin. Either you remember in the form of images or remember it in the form of table. Okay, create a table and you can remember. Formin helps in nucleation plus end. ARP2 bar 3 complex, they will form branches towards the minus end. Thymosin prevents the assembly. That means this is again depolymerization. Propylene speeds the elongation. That means this is polymerization. See, these two are contradictory to each other. Tropomodulin. If you are hearing for the first time, definitely it will confuse you. But if you have already studied and looking over it again, it's a very good revision for you. Tropomodulin prevents assembly and disassembly at minus n. That means it remains neutral. Coffulin, disassembly. Gelsolin, like your catenin, breaks down. Tropomyosin, like your map, stabilizes. Capping protein provides protection. Or I can say assembly and disassembly is prevented. So if you see tropomodulin and capping protein, they are having contradictory function, but contradictory function in the sense in different ends. This is minus end, this is towards the plus end. Then remaining all are the accessory proteins that will help in the attachment to the membrane. Okay, I hope so. Whoever was having the confusion, is it clear to you? Hemlata, dream narrator, Suchitra, Lakshmi, Sunshine. If you are, as I told you, if you are hearing for the first time, this is a confusion. But if you have studied and forgotten, this is a revision. Okay. Now, if you have understood this one, based on this, I have another question for you. So, cellular actin levels can be as high as 100 to 400 micromole. Of this unpolymerized, yes, Hemlata, unpolymerized actin concentration is 50 to 200. Critical concentration for actin polymerization in vitro is 0.2. Now, some of the following proteins inhibit the polymerization of actins in the cells. Thymosin, capping, tropomodulin, XMAP215. Now, you have to identify the inhibitors of actin filaments. Just I have discussed the concept previously. Based on this, another question. Can anyone able to identify who all comes under actin inhibitors? And remember, students, this is very important from examination point of view. Every time question, I mean, specifically in the recent past, I have seen questions coming from here in the form of 4 marks. So according to your comfort zone, either follow the images or follow the table, whichever is convenient for you. Absolutely correct. Gayatri. Red. Anyone else? Sriyushri, very good. Okay, let me show you the answer. The correct answer is 1. Why? Because ABC is considered to be actin inhibitors, but XMAP215 is your microtubule inhibitor. And here they are asking you about the inhibition of actin filaments, not your microtubules. That's the reason ABC is the correct answer. Okay. Next question. Now this is based upon signal sequences. Match the following. This is also a very favorite part of CSIR. Many a times you come across this question in the form of match the following. And the thing is that questions are always asked in the form of this match the following. Lysosome, mitochondria, nucleus, peroxisome with their corresponding amino acid sequences. And simple words, I can say signal sequences. Here also I can see some confusions popping up between option 3 and option 4. Let me tell you one thing. I know that all of you are sure that lysosome is for mannose 6-phosphate. So based on that, you are trying to figure out whether it is option 3 and option 4. I guess this is the concept behind, right? Okay. Let me remove your confusion now. what exactly the answer is. Whoever has given option 3 as the answer, yes, you are absolutely correct. So what is the difference is that, no doubt, whatever you thought is correct. Apart from that, how you can identify these two? See, peroxisome, you are having a tripeptide sequence. PTS1, PTS2, that is SKL sequence, if you remember, we are having. How you can identify mitochondria and nucleus? Both of them are having lysine arginine residue. Well, the difference is when it comes to mitochondria, it should be N-terminus. And when it comes to nucleus, it do not have any terminus. It simply includes only the amino acid, not N or C terminus. Okay, so this is how you need to understand. Now, so this table from Lodish will definitely help you in remembering the things. Please go through it. Important from examination point of view. Then this is based upon the sequences took it from Alberts. Okay, so here you have ER, mitochondria, chloroplast, peroxisome, nucleus. Whether the N-terminus is removed or not, I mean the signal sequence is removed or not, that is also being clearly mentioned. Kindly go through this table, very important. Now, based on that, let me get another question from you. Let me get answer from you, trying to understand whether you understood or not. Find out the correct combination from here. Okay, that's good, Sunshine. You are able to give the answer. Now, based upon the previous concept regarding the vesicles, regarding the organelles, their corresponding sequences, you need to give me this answer. Let me see how many of you have understood the concept behind it. I know. Again, here also you are applying the same logic because lysosome is for mannose 6 phosphate receptors. So you are eliminating option 2 and 3. So you have confusion between 1 and 4. So you have to identify the correct answer, right? If you remember, I have also given you antrograde, retrograde differences also. Based on that also, you can give the answer. Okay, very good. Very, very good. I can see all of you are giving the answers. That means the concepts which I taught you just now is very much clear, right? Very good. So the correct answer over here is 1. So antrograde is a forward movement. Always remember. Clathrin coated vesicle is related to endocytic vesicles. Okay. Cis Golgi, trans Golgi. So for secretion means obviously it is trans Golgi. Okay. So very good. Good to see majority of you has given the correct answers. Okay. Next. Now this is based upon a nuclear transport. RAN GTP levels are higher in the nucleus than cytoplasm. Import receptors can shuttle between nucleus and cytoplasm. NTF2 transports RAN GDP into the cytosol. Export of mRNA is not directly dependent on RAN. tRNA and microRNA export is mediated by exportins. Again, here you have to find out the correct set of statements. Let me see how many of you can give this answer. See, again, the protein targeting related to nucleus is also very important from examination point of view. Remember this point. The reason is that they are asking the questions both in the form of 2 marks as well as in the form of 4 marks. Just modifying that and getting the questions. Even if you go through previous year question papers, have a look onto that. You'll find every time one question comes from nuclear import. Okay, RAN GDP is more in cytosol, you think, Anupam. Okay. Any other answers? Zakia, very good. Is it the question answer from previous one or this one? Sorry, I did not see that. Okay, let me give you the answers. I can see some confusions running on in the answer. Let me show you the answers now. The correct answer over here is A, B, D, E. That means A is correct. RAN GTP are higher in the nucleus. Nuclear import receptors, like that is your karyoferine receptors, if you remember, we are having importing, exporting. They can shuttle between nucleus and cytoplasm. Export of mRNA is not directly dependent on RAN because you are having specific karyoferine responsible for that. tRNA and microRNAs are exported via export teams. That is correct. Now, why this NTF2 is incorrect? Let's find out that. Yes, Suchitra. Statement C is incorrect because NTF2 is the transport factor responsible for import of RAN into the nucleus, not to the cytoplasm. Okay, that's the reason the statement C is incorrect. So this NTRs therefore rely on additional components that refuel the RAN GTP, that is nuclear transport factor 2, that can retrieve the GDP back to the nucleus. See, I'll show you an image. Always remember, RAN GEF is present in the nucleus. If it is present over there, you'll find RAN GTP present, active form. RAN gap is present in the cytoplasm. So RAN GDP also in the cytoplasm. So try to remember this concept. Many a times you get questions from this only because many a times we feel confused. RAN GEF is where? GTP is where? GDP is where? So make this concept clear first. After that, remember this karyoferine receptors. You are having importin receptors, exportin receptors. Okay, so if you remember that, it is going to help you out further. Then understand how the proteins are being imported inside the nucleus. This is your NPC, nuclear pore complex. These are the receptors carrying the cargo, tethering to the filaments, releasing inside the nucleus. Once they are released inside the nucleus, the receptors are recycled back. RAN GTP carries one of the receptors towards the cytoplasm. Exportin carries another receptor towards the cytoplasm. Okay, so that is how the nuclear import and export takes place. I hope so it is clear to all of you. No confusion over there, right? Okay, in that case, find out the answer for this mitochondria related. Actually, this is the organization of 3 proteins. You have to identify it belongs to which compartment, whether it is matrix, inner membrane, intermembrane, or outer membrane. Once again saying, nuclear transport is an important part from an examination point of view. Don't quit that. Okay, Soumya, thank you for confirming. Now find out the answer for this. Is it tough? Is this question tough to all of you? If so, you post it directly so that I can explain you the answer directly. But if you're trying to attempt, well and good. If you find it difficult, do let me know. I will explain you. Because again, we have to look for unit 4 as well. We are still in unit 2. Problem is that both unit 2 and unit 4 are lengthy units equally, and questions also comes very much from both the units. So it becomes practically difficult to cover as a whole, but I'll try my level best to touch at least the important part. Okay, you need explanation? Sure. See, okay, I can see some answers are coming up. Lavanya, very good. Sunshine, Saundarya, very good. Okay, let me explain you how does it works. See, in order to find the answer for this question, you have to understand this image. See, if a protein needs to enter inside the matrix, it should have an MTS attached along with the polypeptide. If it needs to enter inside the inner membrane, either it should have MTS. MTS means matrix targeting sequence. That means after going to matrix, it has to return back to the inner membrane. How? Because it is having some stop transfer sequence. And in another case, they are having an OXA1 protein, which is responsible for exporting the proteins back to the inner membrane. Otherwise, there are some proteins which does not have MTS. They have direct internal sequences. So they will directly tether to the inner membrane. Intermembrane space, one way is by having MTS and the intermembrane space proteases. That means they will be released into the matrix, taken into the inner membrane, and finally to the intermembrane space. Otherwise, they will be directly entering inside the intermembrane space through general import pore. For outer membrane, not much difficulty. Simply, they will be having outer membrane localization sequence so that they can enter inside directly. Suchitra Chatterjee, I guess your confusion is clear now. So this has to be remembered because mitochondria based questions also being asked frequently. Shrivani, you can refer to Lodish for this protein targeting. That's a very good book. To delineate, so next question over here is to delineate the steps in ER transport, a PhD student homogenized pancreatic SNR cells to isolate microsomes, which retain most of the biochemical properties of ER. So for this experiment, the student has planned a number of controls as mentioned below. Treated the microsomes first with detergent, then with protease only, micrococcal nucleus with detergent only. You have to identify the best combination that she can use as a control. Microsomes are the organelles or vesicles that comes out when your endoplasmic reticulum is degraded. Anyone would like to give your answers? I can see it's quite. Is it difficult? Actually, it is not difficult. Why, you know? Let me give you the answer. See, the correct answer over here is A, B, and D. So here, what they are asking is a PhD student has taken pancreatic cells to isolate microsomes. So when you are speaking of microsomes, do you think so? We need nucleases because nucleases is something that is responsible for degrading the DNA. Nucleic acids. So we don't have any role of that. We are we are focusing on ER. So in that case, A, B, and D should be the correct answer. Okay. See, the microsomes are formed from endoplasmic reticulum when the cells are disrupted. You can collect it via differential centrifugation. So detergents are commonly used to lyse the cells, solubilize the membrane proteins and lipids. So in the control, we can use all the three individually or in combination. But when we speak of nucleases, they are attacking nucleosome. That means it is related to chromosome. Here we don't have anything related to chromosome. So you can ignore that. Very easy question, right? 4 marks. Chromatin remodeling. Moving on to chromosome. Chromatin conformation may play more important role than local DNA sequence of the promoter. Histones in nucleosome can undergo different modifications that alter the chromatin architecture. Chromatin remodeling is a regulated process, does not require ATP, and several variants are existing which can replace the standard histones in specific types of chromatin. You have to identify the correct combination. No, detergents are responsible for breaking the membranes, right? So that will be useful for. Proteases can remove the protein portions. So that's why you can use that. But nucleases is an exceptional case that you can ignore. If you're aware of chromatin, now this is the part of your unit 3 also, fundamental process, replication. What is the correct answer? This is also a very easy question, just if you understood one particular thing. I can see many of you are giving the answers correctly. Okay, there is some confusion between, again, option 3 and 4. See, what is the problem over here, you know? The correct answer should be ABD. Always remember, remodeling of chromatin requires the ATP, whether it is sliding, deletion, ejection, replacement, everywhere you require some energy. So ATP must be used. A is correct because definitely chromatin conformation plays a very important role because you are having two stages. One is the condensed stage and another is the open or relaxed stage. Condensed stage is one where it is less accessible to transcription factors. That means no gene expression levels. Open or relaxed stage is one where gene expression is promoted. So the first option is correct. Histones will undergo different modifications like phosphorylation, methylation, ubiquitination, acetylation, etc. Variants exist. Yes, you are having SEN-PA, H2X. So different variants are existing which are responsible for carrying out various processes. That's why this is the correct answer. These are, see, number of variants you can see: SEN-PA, H3T, 2X, AZ, BBT, H2A, H2A.1. And in fact, these are very much important for gene expression because its overexpression or down regulation can cause many diseases, as you can see in this table. Okay. Okay. So these are just mentioning you that among all the histones, H3 and H4 is considered to be highly conserved. Remember this point. And acetylation always leads to activation. Okay, lack of acetylation only repression takes place. Methylation, depending on where exactly the methylation takes place, it either activates or represses it. Remember these 2 points. Important from examination point of view. Okay. Next question. Based upon nucleosome. Histone tetramer in the core of nucleosome comprises of H2A, H2B, H3 and H4. N-terminal tails are believed to stabilize 30 nanometer fiber of nucleosomal DNA by their interaction with adjacent nucleosome. Post-translational modifications of N-terminal tails as well as globular domains modulate the transcriptional events. According to the zigzag model, 30 nanometer fiber, the linker DNA circles around the central axis as the DNA moves from one nucleosome to the next. You have to identify the correct statements among this. Find out. So you can expect in the form of chromatin or you can expect in the form of nucleosomes. Okay, I can see many of you are coming up with the answers. Soumya, Sunshine, Arzu. Yeah, very good, everyone. All of you are giving the correct answers. Happy to see all of them togetherly giving the correct answers. Yes, absolutely correct. So the reason is that the zigzag model, this is not according to zigzag model. This is according to solenoid model. Okay. So that's why D is the incorrect answer over here. Sorry, this has to be, just a second. Answer is 1, B and C. Yeah. Here it is not tetramer. Here it will be octamer, right? I too missed out. Yes, it should be octamer, right? Okay. This is octamer. So that's why A is incorrect. And D is a solenoid model. So we are having B and C. Okay. I hope so this is clear to all of you. So that's the thing I have mentioned. Histones H2A, H2B, all are core histones. 1 and 5 linker histones. All exist as dimers. So 4 dimers combined together forming octameric nucleosome. The last statement was based on solenoid model. Right? So that's why you have the correct answer over here. So literally I too got confused, but no issues. If you read the statements properly, you'll definitely get the answer. So from here, one thing I want to say you, read the statements properly when we are going for examination. Okay. And majority of modifications usually occur in the tail region. Sometimes histone fold domains also undergoes modifications, specifically methylation of lysine 79 of histone H3. Okay. This here the modifications are less. Exactly, sunshine. But in H3, you can see the modification takes place here. Okay. Next. Chromatin condensation is driven by protein complexes called condensins, which are members of a family of structural maintenance of chromatin proteins. Now, they play a key role in the organization of eukaryotic chromosomes. Condensins, along with another family of SMC protein cohesins, contribute to segregation during mitosis. If these cells are treated with an inhibitor of CDK1 phosphorylation before entry into the M phase, which statement is likely to be true? Okay. Sister chromatids are held together by condensins along the entire length of chromosome, or held by cohesins along the entire length. Held by condensins attached to each other only at the centromere or only at the telomere. Find out the correct answer over here. Thank you so much. Struggle. Exactly, Manoj. Just one word, tetramer to octamer. Difference is there. Yes, I can see Gayatri has come up with correct answer. If the cells are treated with the inhibitor of CDK1 phosphorylation before it enters into the M phase, then what should be the answer? Okay, I can see some confusion coming up with option 2 and 3. Let me clear out that. Yes, red also correct. Correct answer is 2. Now, why it is true, you know, we know that CDK is a key regulator of cell cycle, particularly when we are moving from G2 to M phase. And we know that it phosphorylates the target proteins, including the cohesin. What are cohesin? Cohesins are responsible for holding the sister chromatid together from the time of S phase till their separation during the mitosis phase. Right? So, phosphorylation of cohesion by CDK1 will lead to dissolution of chromatid. So, what happens? Chromosome segregation occurs. So, this cleavage of sister chromatids by cohesion is necessary so that the chromatids will be separated. If cells are treated with an inhibitor of CDK1 phosphorylation, then what happens? The phosphorylation will be prevented, cleavage will be inhibited. So what happens? Sister chromatids will attach together by cohesins along the entire length. That is the answer. Okay. So what happens if it is phosphorylation is inhibited? The cohesion will not be removed. They will be attached to the sister chromatids. That's why option 2 is the correct answer. Okay. I hope so it is clear. Whoever are having confusion with option 3, I hope so this clears your doubt. During the cell cycle, entry into the S phase is tightly regulated. Now, why this is so? APCC promotes ubiquitination of S phase and mitotic cyclines, marking them for proteolysis at the mitotic exit. Cyclin B1 helps in the activation of S phase CDKs only in late G1, or as mitotic CDK activity declines, CDC14 phosphatase activates APCC by dephosphorylating CDH1, promoting the formation. Securin keeps the S phase cyclins in inactive state till late G1. Okay. Find out the correct combination out of this. Thank you so much, Zakia. All because you all are responding so beautifully. You are giving the answers so perfectly. That itself makes us active to teach you more, since we are in the last time, like last period of your exams. Okay. What is the correct combination over here? Okay, let me give you the answer. Answer is A and C. Now, why it is A and C, first of all, you have to understand what are the different types of cyclines are. Okay. So you can see, you can remember it as D, E, A, and D. Okay. Remember this format. Then 4, 6, 2, and 1. 4, 6, 2, and 1. First, remember this thing. That itself will clear partially. Next, what you are having, APC. Now, this is bound with CDC20 and CDH1. Now, when it is bound with CDC20 and when it is bound with CDH1, check it out. During mitosis, your CDC20 is the primary activator of APCC. So, that will be responsible for regulating the some mitotic proteins. As the cell exits the mitosis, it enters the next cell cycle, that is G1 phase. At that stage, CDH1 will be combined. So CDH1, even though it is present, it will be inactive by the phosphorylation of CDKs. But when we enter into the late phase of mitosis, CDK level declines. Due to this decline, phosphatase will be activated. Now, this phosphatase will remove the phosphate from CDH1, leading to the activation of CDH1. This concept has to be understood. Next, combining these two concepts, you can easily get the answer as A and C. And securin, though, it does not work in the G1 phase, right? Usually it is related to the G2 phase, so you cannot use it over here. B, as I already told you, this one, right? So that's why A and C are the correct answers. Next, cell cycle can also be asked in the form of match the following also. See, recently, last exam, this match the following came in the exam. Are you able to identify the correct match? CDK1, APC, CDC20, CDK2, Aurora B. I guess you are able to identify what is Aurora A and B function and all. When it comes to cell cycle, you have to be very careful by picking up proteins name. Proteins functions are very important. So when you are studying cell cycle, make sure that the functional aspects of proteins, the concept behind the functions of each and every protein has to be clear. cohesin, condensin, securin, separase, aurora AB, ATM, ATR. A lot of proteins are there. So you have to be very clear with that. Yes, I can see many of you giving the answers. Absolutely correct, because I can see every one of you has given the correct answer. So A to 3, CDK1 helps in mitotic entry. B to 4, that is anaphase onset. Just now only I have discussed with you. C to 2, CDK2, S phase entry. This also just now discussed. Aurora B, they are helping in cytokinesis. You might be aware of Aurora A and B, right? The Aurora A is basically contributing to entry inside the mitosis, whereas B helping in cytokinesis. How? Helping in the contraction of contractile ring, facilitating cleavage furrow ingression, as well as completion of cell division. Okay. Next. So this is just an example. Like in the last exam, this question came from microbial physiology based upon the extremophiles along with their environment. This is a very easy question. I guess anyone can answer. Depending upon the the environment, the names are being given. Are you able to get this answer? You can expect microbial physiology question from here, as well as sometimes you can also get a numerical question also. So just remember the formula over there. Okay. Then the significance of different phases of microbial growth. That also is important from examination point of view. Okay. So this is just a sample to show you last time this question came. Correct answer is answer 3. I can see some of you has already given the answer. Yes. Psychrophile means cold. Thermophile means heat. Alkalifile, you can alkaline environment. Piezophile is related to pressure. Halophile is related to salinity. Okay. Now quickly moving on to unit 4, and the very first question from unit 4 is signal transduction pathway involved in glycogen metabolism is triggered in the liver by the hormone epinephrine. Now this includes these many steps. You have to identify the right order in which the steps are activated. G protein, protein kinase A activation, second messenger generation, AC activation. So just because I'm running out of time, I quickly jumped into unit 4 so that at least I can touch some of the important parts from unit 4. I hope so. I'm fine. You all are fine with it. Unit 2 also maximum I try to touch the important aspects, because basically when I say protein targeting, chromatin structure, cell cycle, very important from examination point of view. Then minor ways, solute transport, membrane structure, solute transport, microbial physiology, etc. Okay. Yes, I can see some answers coming up, but some miscellaneous answers coming. There is some confusion between 1 and 4. Okay, let me give you the answers because I can see much confusions coming up. Correct answer is ADCB. That means first your G protein has to be activated. So then comes your effector protein, then comes your second messenger, then comes your protein kinase. So remember, a background I'm giving, when a ligand binds to the receptor, AC gets activated, that is effector protein gets activated, which leads to second messenger, which gives rise to protein kinases. The kinase will phosphorylate the target proteins to generate cellular response, and finally it leads to termination. That is what the thing is. So if you look into the image, you can see if it is in the resting phase, this is what the concept, inactive forms, and GDP bound G protein. If hormone binds, Receptor undergoes conformational change. G protein binds to it. When it binds, GDP is released. GTP goes inside. At that stage, alpha, beta, gamma will be separated. This will go and bind to the effector protein, releasing the activating the signal transduction pathway. If you want to terminate it, all you need to do is hydrolyze this GTP. So what happens? You will get a trimeric G protein like this. Okay. So this is your GPCR. Second, G protein is active when it is bound with GTP, and terminal phosphate is bound with glycine and threonine. G protein is inactive if the terminal phosphate is degraded, and that is GDP bound form. Okay. So this is your pathway is. The same thing what I explained you over here. Epinephrine binds to the receptor. Your ATP AC activates the second messengers. Then it activates the protein kinases. Then the corresponding enzymes will be activated. Glucose will be produced. This will increase the blood glucose level. Okay. I hope so there is no confusion over here. Now, this is a question based upon the membrane potential. A potential difference of minus 70 MV between inside and outside of a single axonal membrane in resting condition may be recorded by suitable electrodes and amplifiers. So, here are some statements. You have to find out the correct combination. RMP is close to equilibrium of sodium ions. Unequal distribution of diffusible ions across the RMP, across the axonal membrane for RMP. Axonal membrane must be permeable to one or more species of ions for the RMP. Concentration of sodium and potassium across the membrane is required for RMP is dependent on activity of sodium potassium ATPase. Impermeable proteins do not affect the diffusion of diffusible ions across the membrane, which is required for RMP. Then, sodium potassium ATPase in the axonal membrane is essential for RMP, but it is not electrogenic. Find out the correct combination over here. What you need to study. So others, please focus over here and try to give the answer. Okay, I can see Soumya has given the answer. Struggle. In the last 2 days, you cannot do anything. You cannot take any new topic. All you can do is revise the existing topics. Whichever topic you have already completed, you might have tables or flowcharts or images of that. Just swipe those pages, because you cannot, literally speaking, you cannot take any topic at this junction. And don't be tensed. Whatever you have studied is already there in your mind. All you need to do is brush up. And that's the reason, if you have attended all the series for the past 4 days, you might have come across many clearance from many topics. Okay, so don't worry. Yes, life science, clearing a CSIR NET exam is not like clearing an MSc or BSc exam where you will be sitting for one night and you will revise, gulp up everything. That is practically difficult, because as I already say you, life science, like many of you are my own students whom I taught. I always say life science is like an ocean. It's like a sea, endless. But you have to pick only those things which you are comfortable with, not everything from that sea. Okay. So 1 day or 2 day for revision is not enough. 1 week revision is there, but you are already in the revision way. For the past 4 days, you are already revising the topics. Then in the hand, you are having 2 more days. Just pick up small topics which you have already studied and try focusing on that. Okay. Don't worry. Don't be too much tensed. Anupam, struggle, and to all of you who are facing the same thing. Believe in yourself that yes, you can do that. Yes, I can see some answers coming up for this. Now, this is related to what you know, ligand gated ion channel. If you remember in the class which I taught you. So, what is the correct answer over here? Answer 2, B, C, and D. Of course, RMP is minus 70 MV. And sodium ion saturation is not the same. So, definitely this is incorrect. Unequal distribution has to be maintained. Then only the membrane becomes inside negative and outside positive. If you remember, 3 sodium to 2 potassium. Axonal membrane to permeable to one or more species of ions. Obviously, sodium and potassium. Concentration is dependent on, as I told you, sodium, potassium. See, with this one concept, you can easily get the answers. If you are confident that these 3 statements are true, then E and F is hardly mattering. It is saying that it is not electrogenic. How it won't be electrogenic? It is electrogenic. It is generating electric volts, ligand gated ion channels. So obviously this is incorrect. This is also incorrect. Okay. Don't be panicked by looking into the question, because the answer is there inside you. All you need to do is read the question thoroughly, patiently. Important thing is that patience. If you read the question patiently, you'll get this answer for sure. Okay. I hope so. I cleared your doubt, whoever was asking the answer. Okay, many of you has given 2 as the answer, right? Very good. So this is the events. This is your minus 70 MV. Upon getting a stimulus, you reach a threshold. Then sodium ions will come inside, call it as depolarization. Potassium ions go outside, you call it as repolarization. When it goes beyond the RMP, you call it as hyperpolarization, right? Absolute refractory period, when you try to disturb this action potential without any completion, it won't start. Relative refractory period means when you're giving a stronger stimulus after the completion of first stimulus, you are calling it as a relative refractory period. Okay. The action potential and resting membrane potential is clear to all of you. I hope so it is clear. From this, and the question which I had asked you was a last year question. Okay. Okay, then. So now there is a match the following for you. Find out the correct combination. So after going through these questions, you are feeling nervous or you're feeling confident? After completion of second unit and we are in the fourth unit, do you feel nervous or do you feel confident? I can see many of you are giving the answers. That's really good to hear from you, Anupam, Gayatri, Saundarya, because these sessions are done not to generate scare or what I can say tension inside you. This series was conducted to remove those tensions and to boost your confidence when you are approaching the exam. And seeing that, it really makes me feel good. You should not be nervous. You should be confident that I am able to give the answers. Keep this positive attitude with yourself. Okay, let's time to show the answer now. All of you has given the correct answer. Very good. Hemlata, why are you so nervous? Because I can see many of the answers you have given correctly. So receptor tyrosine kinase, blindly you can say it is related to insulin signaling. So you can easily pick up 1 and 3 can be eliminated, 2 and 4. Next, Q to 4. CGMP is related to GC. GTPase activating protein. Just now only I said if gap is there, it results in hydrolysis. Hydrolysis means inactivation. Okay. And finally, nuclear receptors. Example is thyroid hormone. Now, this is an example of class 2 receptor whose receptor resides in the nucleus present within the DNA sequences directly. Unlike the other one, which is class 1 present in the cytoplasm. Okay. Okay. Next combination. Don't feel nervous, Hemlata and Soumya. Feel confident and take the count how many answers you have given correctly. Okay. And in the exam hall, you will definitely able to recollect all the questions, answers, concept. I will not say question answers, concept. Because end of the day, it is your concept that will help you to get the answer. Yeah. Coming back to our question. Exactly, Sunshine. That's what I wanted to hear from you. Yes. Find out the correct combination. And when I speak of cell signaling, mainly 4 marks, you can always expect a combination statement or you can expect this kind of match the following. Last time they have provided you one image of receptor tyrosine kinase and they had asked you the events that occur. So basically direct questions only comes from here. Okay. Majority of you I can see giving the correct answers. Very good. Soundarya, Arzu, Red, Kavya. See, how does it works? The correct answer is 1. How? First, take one thing confident inside. You know that insulin is an example of receptor with intrinsic kinase activity. So you can blindly say this is the answer. So if you got D as 4, there is no competition over here. You can easily click on to option 1. Then if you speak of interferons, like we have erythropoietin, you remember it follows JAK-STAT pathway. And in the JAK-STAT pathway only, you are having cytosolic tyrosine kinases. They do not have intrinsic kinase. They have cytosolic kinase. So again, B to 2, you got another confident. So this is basically a question where you can easily get an answer with just one answer if you're confident of. And I firmly believe that everyone is aware of insulin, right? Then serotonin is GPCR. Glycine is responsible for opening of specific ion channels. Even though you're not so sure of A and C, but you have to be confident on B and D. So you can easily get the answers. Right? Very good. Now, this is a question which comes very rarely regarding bacterial chemotaxis. You have to identify the incorrect statement from here. Receptors are dimeric proteins that bind to specific attractants and repellents on the outside of the membrane. Cytoplasmic tail is stably associated with a histidine kinase known as key A via an adapter protein key W. Receptor and its associated proteins are all clustered at one end of the cell. Binding of attractants increases the activity of receptor while repellent decreases the activity of receptor. Now find out the correct answer from here. Speaking regarding chemical like bacterial chemotaxis, sometimes you can get question from here, but not always. Just try to brush up your concept. You never know, you can expect, because this question came some year back. I mean 2 to 3 years back. But right now you can expect because it's been some time. Okay, very good. Anupam, Soumya, Manoj, Kavya, Suchitra, Sunshine for giving the correct answers. What about others? Yes, you are correct. 4 is the incorrect answer. You know why? Any of these questions I can you cannot expect, but any of these related questions can definitely come in your exam, Zakia, because CSIR, you know their rule, right? They never repeat the questions, but they can ask the questions relevant to that. Okay, definitely feel confident, you will definitely come across these questions which we are discussing today. Believe in that. Okay, so bacteria is having two component system. First component is the histidine kinase, another one is the aspartate. So when this attractant goes and binds to the receptor, kinase will be activated, transferring the phosphate, and upon phosphorylation it will go towards the flagellar motor and helps in the rotation, depending upon whether attractant or repellent is there. I will come to that point, Anupam. Yes, I'll come to that point. Exactly, Suchitra. See, we have counterclockwise direction if attractant is there. That means if food is available. If the clockwise direction, if we have repellent is present. Tumbling when all the flagella is not coordinating. But here all the flagellas are coordinating. Okay. Now what happens, when a repellent goes and binds to the receptor is active. This information is passed from key W to key A, which undergoes hydrolysis, transferring the phosphate to key Y. Phosphorylated key Y helps in the flagellar motor in the clockwise rotation, resulting in tumbling. On the other hand, if attractant goes and binds to the receptor, what happens? This activity will be reduced. Phosphorylation will be reduced. Not only that, whatever the phosphorylated key Y was there, will be acted on by key Z, and they will remove the phosphate group and make it as a dephosphorylated key Y. This will help in counterclockwise direction, helps in running or swimming. That is the concept over here. Now, if you apply this concept in this image, you will understand better. If you are bound with a repellent bound to a receptor, key R will add methyl group to the receptor, making it activated. So, key A upon activation adds phosphate to key B and key Y, tumbling. On the other hand, if attractant goes and binds to the receptor, this phosphorylated key Y will remove the methyl group as methanol. So what happens? The phosphorylation activity will be reduced. So here also reduction, and this leads to running or swimming. I hope so this concept is clear. Anupam and Suchitra, remember this concept. Okay. Next question. Now this is with respect to CGMP signaling. ANF causes natriuresis and diuresis by interacting with membrane bound form of GC. Now this A and D, you can see this is membrane bound, this is soluble form. One of them has to be correct. Nitroglycerin causes smooth muscle relaxation and vasodilation by interacting with soluble form. Prusside causes the same thing but bound with membrane. So from this, what I can say, among A and D, one has to be correct, and among B and C, one has to be correct. Now find out the correct combination out of this. Yes, Anupam, I hope so you have received the explanation for the question you had asked me. Suchitra also. Okay, absolutely correct. Hemlata, Krishna, red. Anupam, red. Shana Nazreen, Soundarya. Shana Nazreen, have a rethink, because A and D will not come together. I can see all of you are giving the correct answers. Yes. If this nitroprusside was given soluble form, it would have been the correct answer. Since it is given membrane bound, so it became the incorrect answer. Okay. So these are the two forms of GC. One is membrane spanning, another is soluble nitric oxide activated. Membrane spanning, you have example of ANF, guanylene and endotoxin. Now this is nitric oxide activated. So from the arginine, you will get the nitric oxide, and that will help in the activation of this GC. Then it will help in the opening of specific ion channels, smooth muscle relaxation in heart, blood vessels, etc. Okay. Now this is with respect to GPCR GT pathway. Here are some explanations. Upon prolonged illumination, activated rhodopsin does not activate transducin, and hence the vision is impaired. So this were the explanation. Most of the activated rhodopsin gets phosphorylated and is unable to activate transducin, or gets dephosphorylated and unable to activate transducin. So either of this has to be correct. Arrestin further interacts with phosphorylated rhodopsin, or it interacts with dephosphorylated. Find out the correct combination over here. GT pathway. If you are able to recollect the pathway, what happens in the light, what happens in the dark, you'll be able to get the answer. What should be the correct answer over here? Yes, Manoj Soundarya has given the answer. Red also gave the answer. Okay, let me show you the answer now. The correct answer over here is B. Now, why it is B? Most of the activated rhodopsin gets dephosphorylated. So, once it is dephosphorylated, what happens? It is unable to activate the transducin. And arrestin further interacts with dephosphorylated rhodopsin. Actually, in this case, what should be the correct answer? Do you think B is the correct answer? Everyone? Are you able to correlate whether it is correct or not? It has to be which one? Okay, let me first give you the explanation how exactly it happens. See, during the dark, you are having CGMP is active, cation channels are open, action potential takes place. This results in hypopolarization, thereby releasing the inhibitory neurotransmitters. And bipolar and ganglionic cells are inactive, and that's why no vision. So here what they have asked, due to prolonged illumination, transducin is inactivated. I think partially you understood now. Prolonged illumination does not activate the transducin. Next, in the light, rod cells are inactive. So CGMP gets converted to GMP. Cation channels close, no action potential, hyperpolarization, excitatory neurotransmitters leading to vision. You're getting me, right? Next, if you look into the image over here, check out the final step. So we have light phase, dark phase. After that, adapter phase. Now, in this phase, what happens? Rhodopsin kinase phosphorylates the rhodopsin. So this is phosphorylated. This is phosphorylated. Into that, arrestin comes and binds. So what happens? Rhodopsin is inactivated. Exactly, Sunshine, you got it. Next, after some time, arrestin will be dissociated and rhodopsin will be dephosphorylated. So what are these two? These are the proteins, that is all cis retinal, all trans retinal, 11 cis retinal. So trans retinal is going out and cis retinal is coming in. Now the rhodopsin is ready for another phototransduction cycle. Did you get the point, everyone? You are able to link this to that question? Yes? Can I get a reply from all of you? Are you able to connect this to this question? Most of the activated rhodopsin gets the... Okay, Sandhya, Saundarya got it? I hope so. Everyone got it? Yes? Very good. So that's the fact. Okay, fine. So no need of further explanation. Next, we are moving on to 4C, cellular communication. Cellular communication, you can expect always match the following majority of cases. Okay, so these are cell adhesion molecules. You have to identify the correct match. Exactly, Arzu, that I can relate very well. See, for that, that's what I said you initially. You have to be patient. Patience will give you the correct answers. So you have to be very much patient when you are going for attending any competitive exam. That will definitely help you. Okay, I can see all of you are given the correct answers. Absolutely correct. So remember this table, which is taken from Cooper. Selectenes, if they are bound with carbohydrates, there won't be any stable junctions. But integrins, if they are bound with ECM proteins, you can form a stable junction like focal adhesion and hemidesmosomes. Similarly, cadherin also promote stable junctions like adherence junctions and desmosomes. Okay. And remember, all the adhesion molecules requires divalent cations for their activation. Another type of question. See, majority of cases you'll find this match the following kind of questions only. Okay, you may go through this question because these are very direct questions. You can easily get the answers. I wanted to explain you the questions related to apoptosis and cancer in details. Just running out of time, so only I'm going on quickly because these are very direct. If you know what are these proteins, you can easily get the answers out of this. See, tight junctions, gap junctions. This was the question came last time. Very easy questions. You can easily get the answers out of this. Okay, no problem with that. Please look into this table taken from Alberts and this table taken from Lodish. If you're reading this, getting the questions from any of those junctions and proteins, you'll easily fetch a 4 marks out of this. Okay. Okay. Or else sometimes you can expect experimental questions like this, like see, cadherin's medium calcium dependent cell adhesion and plays an important role in embryonic development by changing the adhesive properties of cell. Aggregation of nerve cells to form an epithelium is correlated with appearance of N-cadherins on cell surface and vice versa. NCAM involved with adhesive interactions. So you have to study the mutations of these two proteins. So two sets of mice were generated. Set A mice with mutation in N-cadherin and set B with mutation in NCAM. So what will be the ultimatum? In this, the answer should be option 2. Why? Because you know that cadherin is required for early embryogenic development. That's why the mice of set A will die early in the development. But NCAM is required for proper adhesion interaction. That's the reason, no doubt, the set B mice will develop normally but will have abnormalities related to nervous system because this is a neuronal cam. Okay. Huh. Now, I'm just taking out some experimental questions. Cervical cancers were untreated and treated with a compound X. Now, what is this X? It is an anti-cancer drug. The cell extracts were analyzed by immunoblotting for the levels of specific markers as indicated by thick band thickness. This is the result. You can see here there is a thick band FADD. Here there is a thick band caspase 8. Caspase 3 is cleaved. P53 is cleaved. Now you have to identify the best option that describes the action X. So you saw this image, right? These are the thick bands you got. Now check out the options over here. Take it as option 1, 2, 3 and 4. Compound X induced cell death via intrinsic pathway. Second one is via extrinsic pathway. Third is by reducing the expression of BAX in P53 dependent manner, or activating the death domain together with increasing the pro-apoptotic proteins. According to you, what is the correct answer over here? Very easy question. If I will tell you the answer, this is a very easy question. Anyone? No? See, I'm giving you the answer. The thing is that you have to be clear with intrinsic, extrinsic pathway. So if you are confident that extrinsic pathway is where your caspase 8 is activated, you can easily click on to the answer over here. What is the answer? It induced cell death via extrinsic pathway by inducing the Fas ligand associated death domain. How? See. Your extrinsic pathway is the one where caspase 8 is activated. Intrinsic pathway is the one where caspase 9 is activated. So we have already seen in the image over here that caspase 8 is having the thick band. Along with that, FAD is also having the thick band. So blindly we can say that option 2 is the correct answer. And obviously P53 has to be, because we are using the anti-cancer drug over here. So that's why option 2 is the correct answer over here. I hope so you are getting me. Can you able to identify the best? Which one indicates the apoptosis over here? Analytical experimental questions are always expected from cancer. Can anyone identify the correct flow chart that leads to cell death pathway? No one? For this, you need to understand this one. If SED9 is active, SED4 is inhibited, SED3 is inhibited, this leads to cell survival. But if SED9 is inhibited, SED4 is active, SED3 is active, this leads to cell death. So this thing, you can find it over here. Now this is an ligand that prevents the SED9 attack, which in turn activates the SED4, which in turn activates the SED3, and this leads to apoptosis. So option 4 is the correct answer. Okay. Then sometimes you can also get questions in this form, that is match the following. So here you have to understand the terminologies like what is a leukemia, what is a sarcoma, carcinoma, melanoma. With this, you can easily get the answer. So this is our direct questions. You can relate to it quickly unless and until you know what is the definitions are. Okay. So this also you can go through it later because these are direct questions. Gradient of oxygen available to the cells at inner regions of tumor tissue environment is typically low. That creates a hypoxic microenvironment. If enough oxygen is supplied to the cancer cells residing in hypoxic microenvironment, which of the following processes will not occur? One thing, if there is something known as Warburg effect, put a focus on that. What it is saying. See, do you know what is the Warburg effect is? Are you aware of Warburg effect? See, Warburg effect means in spite of the presence of oxygen, there will be increase in the level of glucose uptake and production of lactate formation takes place. That means presence of oxygen present or not, it hardly matters. Warburg effect means there will be increase in the level of glycolysis irrespective of presence and absence of oxygen. So that is what here it is given will be reversed, and conversion of glucose to lactate will not take place as sufficient oxygen will be available for oxidative oxidation. That is an incorrect answer. Okay. This is the difference. So basically, it prefers to undergo glycolysis and further goes for lactate fermentation. Why? Because here they can generate NAD plus, and this NAD plus can be utilized by glycolysis to produce carbon skeletons. Carbon skeletons for necessary biomolecules for the survival of the tumor cells. Okay, that is the thing. Then this is also yet a graphical questions which you can easily get the answer. As you can see here, drugs have arrested the growth of cells at G1 phase. How you came to know? CDK6 cyclin D1. Then cleaved PARP is an indicator of a cell that has undergone apoptosis. So that becomes your answer. JAK-STAT pathway. If it is targeted, that's why you are having thin bands over here. Drug led to apoptosis, that becomes the marker. But we cannot say that whether it is extrinsic or mitochondrial or whatever. So ignore that. So that is another way of asking the questions. Now here you have to compare the difference between the graph of VEGF twist and cyclin D1, which determines the metastatic cancer cell. To understand this, you have to understand the function of these three. VEGF is responsible for angiogenesis. Twist is responsible for EMT, that is epithelial to mesenchymal transition. And overexpression of cyclin D1 is always seen in case of cancer. So obviously, if you look into the graph, the correct answer over here will be this one. Okay, because cyclin and twist are equally important. All are equally important. So these are the explanations over here. EMT is the property of cells losing adhesion and gaining migration, highly useful in pluripotency of stem cells. VEGF is for angiogenesis. Twist is for carrying out EMT, and cyclin D1 is always expressed. So that's the reason the correct answer is graph 4. Okay, students, so we are running out of time today. I hope so this has helped you somewhere on how to improve yourself or how to approach a particular question when it comes to unit 2 and unit 4. I heartily wish you all the best of luck, like wishing you all the best wishes. I am short of words actually. Best of luck, everyone, and you all have the potential to clear this exam. Take a positive attitude with you always, and read the questions properly whenever you are in the examination hall. As I told you already, you have to be very much patient enough to read it properly. Okay, so thank you so much, each one of you, and once again wishing you best of luck. Thank you.